Welcome to the Neurostimulation podcast. I'm Dr. Michael Passmore, Clinical Associate Professor in the Department of Psychiatry at the University of British Columbia in Vancouver, Canada. The Neurostimulation podcast is about exploring the fascinating world of neuroscience and clinical neurostimulation in particular. Every week we have interesting conversations with leaders in the field. We have conversations with experts in general health and wellness. And from time to time, we focus in on research studies and information that is included in important textbooks in the area of clinical neurostimulation. In today's episode, we're going to continue in our exploration of the textbook called Practical Guide to Transcranial Direct Current Stimulation. It's going to be another exciting exploration into the fascinating mechanisms behind tDCS. What's really happening inside the brain when we deliver that tiny current? Stay tuned to find out. So tDCS, or Transcranial Direct Current Stimulation, is a non-invasive brain stimulation technique that delivers a low-level electrical current to targeted brain regions through electrodes on the skull. While the method is deceptively simple, typically involving only 1 to 2 milliamperes of direct electrical current, the science behind its effects on brain function is anything but basic. So why care about it? Because tDCS has been shown to modulate mood, cognition, motor performance, and can even enhance recovery from certain neurological conditions. But understanding its full potential hinges on knowing the biological mechanisms that underlie both its acute effects, those that occur during and shortly after stimulation, and the after effects, which may last for hours or even days to weeks afterwards. Let's start with some fundamentals. So when tDCS is applied, it seems to alter the transmembrane potential of neurons in a polarity-dependent way. Anodal stimulation, the positive electrode, generally depolarizes neurons, bringing them closer to firing, whereas cathodal stimulation, the negative polarity, tends to hyperpolarize neurons, making them less likely to fire. But it's not always that simple. The direction of current flow, the neuronal orientation, and the pattern of cortical folding of the outer part, the cortex of the brain, all play a role. Neurons that are aligned in parallel to the electrical field will be affected differently than those aligned in a perpendicular fashion to the electrical field. This is why brain modeling is crucial to understanding individual responses. What about the acute effects? This appears to be more than just influencing neuronal firing rates. During stimulation, tDCS can change a number of things. First is the spontaneous neuronal activity. Second is the responsiveness of neurons to sensory input. And third is the firing thresholds of neurons. For example, studies using single-cell recordings of neurons in animal studies and in brain slice preparations have shown that direct current fields as small as 1 volt per meter can significantly modulate neuronal excitability without triggering action potentials themselves. This mechanism is known as subthreshold modulation, meaning that neurons are kind of nudged toward or away from firing, depending on the current that's being applied. This type of modulation can shift the balance of excitatory and inhibitory signaling, which might explain why tDCS can reduce symptoms in depression or enhance learning in applications to target cognitive issues. Now let's look at the after effects. What might we be able to say about lasting change through neuroplasticity? One of the most intriguing aspects of tDCS is the after effect. Even after the current is turned off, neural changes appear to persist. Why is that? The answer lies in neuroplasticity. Key mechanisms appear to include long-term potentiation, which is important for memory formation, as well as long-term depression-like processes, modulation of NMDA receptor activity, particularly under anodal tDCS, involvement of voltage-gated calcium channels, especially of the L type, and signaling of brain-derived neurotrophic factor, or BDNF. Animal studies have shown that tDCS can modulate gene expression and alter synaptic strength. And these changes are thought to underlie learning and memory, and they're why tDCS is being used in areas like stroke rehabilitation and cognitive enhancement research. Now let's consider beyond neurons and look at some of the important aspects of the microarchitecture of brain tissue, things like glial cells, the vasculature, and how that all fits together to produce the network effects in the brain. Recent research has moved beyond a neuron-only model in terms of the effect of tDCS. It is increasingly recognized that tDCS may also affect cells like astrocytes, which regulate the neurotransmitter uptake and ion balance, microglia, which potentially involves an alteration of inflammation or neuroprotective mechanisms, and endothelial cells in the vasculature of the brain, which influence things like neurovascular coupling, cerebral blood flow, and the blood-brain barrier. Then there are these network-level effects. So tDCS appears to be able to modulate things like functional connectivity, not just at the stimulation site, but also across resting state networks like the default mode network or executive control networks. So this could explain how targeting the dorsolateral prefrontal cortex, or DLPFC in particular, with tDCS can improve things like working memory or alleviate depressive symptoms, in part by modulating broader brain circuits. Now let's consider the complexity and the importance of personalized treatment in order to address individual differences in the clinic. tDCS is not a one-size-fits-all treatment. Variability arises due to factors like skull and scalp thickness from person to person, cortical folding and subtle differences in brain anatomy, as well as neurotransmitter system differences, and the ongoing brain state at the time of stimulation. These factors are why computational modeling and personalized dosing are increasingly recognized as very important. The new research emerging on high-definition tDCS, or HD tDCS, and MRI-guided targeting is aiming to improve the focality and reliability of outcomes so that the treatment can be delivered in a more personalized manner. So what are the clinical implications and some directions for future research? Well, understanding all of these mechanisms isn't just academic, it's highly relevant to clinical practice. If we know how tDCS alters neuronal excitability and neuroplasticity, we can do a few things. First is fine-tune treatment protocols for specific conditions like depression, stroke, or chronic pain conditions. Second is combine tDCS with things like cognitive training or physiotherapy for synergistic effects. Third is to design better devices that optimize current delivery based on individual anatomy and clinical needs. Yet there are still questions such as what's the best dose for a given condition? How do we predict responders versus non-responders? And can we make tDCS more consistent in terms of effectiveness and tolerability across individuals? It's clear that tDCS is a powerful neuromodulation tool with both promise and complexity. So by continuing to uncover its underlying mechanisms, from cellular polarization to synaptic plasticity and network dynamics, we're moving closer to harnessing its full potential in both the clinic and the lab. Thanks again for joining me on this deep dive into the science of tDCS. If you enjoyed today's episode, don't forget to like and subscribe, leave a comment or a question or a review in the comment section below. And remember to share this with a colleague or a family member or a friend, really anyone that you think might be interested in neuroscience or clinical neurostimulation. Thanks again for joining me on the Neurostimulation podcast today. I really appreciate your interest, your time, and your attention. Tune in next time where we explore another interesting topic in the world of neuroscience and clinical neurostimulation. Until next time, be well and stay curious.